Hendra virus infection dynamics in Australian fruit bats. The well-known Hendra virus emerged in 1994 in Australia. This zoonotic agent was first noted when a prominent Queensland horse trainer and 14 of his horses fell ill and died soon after. At the time of the article publishing, there has been 7 noted human cases and 45 equine cases of the virus, due to the virus being spilled from its reservoir. The virus infects horses and humans via a wildlife reservoir. In the case of Hendra, this is through fruit bats, also commonly known as flying foxes. These fruit bats are a natural host of the virus and are asymptomatically affected. To provide further information on the infection dynamics of the Hendra virus, a 3-year longitudinal study was undertaken using a reverse transcription polymerase chain reaction, also known as RT-PCR. This technique is commonly used to detect gene expression levels. In this case, this is the Hendra virus genome in pooled urine samples that were collected from underneath flying fox roosts. The objective of this were to investigate infection occurrence and frequency in flying fox populations and to investigate the genetic diversity of the Hendra virus in the flying fox populations. 3 different roost sites were identified in 3 geographically differing sampling locations. These locations were all greater than 1,500 kilometres apart. One was in southeast Queensland, one in far north Queensland, and the other in the Northern Territory. These locations were visited every 4 to 8 weeks. In the late afternoon, at the selected roost, plastic sheeting was placed under the trees where the flying foxes were identified to be roosting. The following morning at around sunrise, pooled urine samples were collected from each and held on wet ice. A total of 1,672 pooled urine samples from 67 different sampling events over a 3-year period were collected and tested. There were 1,138 samples from southeast Queensland, 320 from far north Queensland, and 214 from Northern Territory. 25% of these sampling events and 2.5% of the urine samples yielded detections. The proportion of positive samples was very highly associated with urine location. These findings indicated that Hendra virus excretion occurs periodically rather than continuously, and in geographically dispersed flying fox populations in the state of Queensland. The lack of any detection in the Northern Territory suggested prevalence possibly varies across the range of flying foxes in Australia. The findings suggest that flying foxes can excrete virus at any time of the year. Why this does not correlate with the seasonal clustering of Hendra virus that has been indicated in horses and associated humans. 70% of Hendra virus infections have occurred in June to October. This must mean that other factors other than the presence of the virus can affect virus infection in horses and humans. Therefore, the identification of these factors will strengthen risk minimisation strategies for horses and ultimately humans.